Welcome, I'm Dr. Marina Gole, Senior Scientist in the GMP Compliance Department at Franz Ziel GmbH. Before this role, I was head of aseptic processing, so I have worked extensively with bio-decontamination and aseptic manufacturing processes. Today, we are discussing VHP bio-decontamination with our subject matter expert James Drinkwater. James is head of GMP Compliance at Franz Ziel GmbH, an honorary member of PHSS, co-lead of the Annex 1 implementation and aseptic processing focus groups, and a board member of the PDA UK chapter. The vaporized hydrogen peroxide process, or simply VHP, has been used for many years in pharmaceutical manufacturing. It is proven to deliver a 6-log sporicidal efficacy and is widely applied in isolators, material transfers and clean rooms. But regulators are now starting to challenge certain aspects of this established process, especially around process understanding and validation. So today we'll be exploring some of those challenges as well as the knowledge gaps. In this video series, we'll look at topics such as the strengths and limitations of VHP bio-decontamination, comparisons with dry fogging and biocompatibility, cycle validity, process monitoring and optimized cycle times, and managing BI quality variances, including the challenge of so-called rogue BIs. So let's begin by getting the perspective of our SME. James, my first question for you is, what are the advantages of hydrogen peroxide vapor biodecontamination that have established this process as a primary method of gaseous biodecontamination?
Hydrogen peroxide is a powerful oxidizing agent with broad spectrum efficacy. Via free radical attack, that 6-log sporicidal reduction can be achieved, and that can be proven with biological and enzymatic indicators. It makes the ideal choice for automated biodecontamination in barrier technologies to establish those essential grade A ISO 5 conditions. It also has applications in, as you mentioned, clean rooms, material transfers into the clean rooms in combinations with open RABs. The applications are wide. We also have the significant advantage of hydrogen peroxide, apart from its efficacy, that you can safely discharge it to the environment because it breaks down to oxygen and water. So a very manageable process for safety.
Thank you, James, for sharing those insights into the advantages of VHP biodecontamination. Let us now turn to a topic that has been raised by regulators. The MHRA, the Medicines and Healthcare Products Regulatory Agency for UK, released a blog on the fragility of VHP. But is vaporized hydrogen peroxide really fragile, or are we simply dealing with limitations that need to be better understood and managed?
Thank you, Marina. We understand from the MHRA that the reason that they released the blog was to start the industry thinking and increasing the knowledge and understanding around the science of this VHP process. This is essential so that the efficacy was not overclaimed and the limitations can be understood and managed. Process understanding is essential to assure successful cycle outcome, thus meeting requirements of that 6-log sporicidal efficacy. What we consider as the key limitation is that VHP is not a penetrative process and not a true sterilization process like moist heat, dry heat, gamma radiation, all detailed in pharmacopoeias. VHP is not detailed as a sterilization process. As such, we have the limitations that it's a surface treatment and the limitations are penetration. So penetration of soiling, we have to make sure soiling is removed. We have to have exposure of all surfaces. We also have to consider the science behind this biphasic process. It has a Gaussian phase, it has a condensed vapor phase, and there's process variables with each. We have to understand and manage those process variables. We also have to increase monitoring to manage the biphasic process, so we add validity, which we'll talk about in another blog. We have focused on exactly these challenges. We really are managing the limitations and controlling the variables to the best possible.
Thank you, James. And don't forget to join us for the next in the series of insights where we compare VHP and dry fog biodecontamination processes and biological product biocompatibility with residual endpoints of VHP cycles. So see you next time.